Okay, so here we are. We have generated an action potential on the sarcolemma of the myofiber. Something I want to mention before I continue the story is that this local depolarization at the membrane is called an end plate potential. End plate potential. The end plate is the specific region of the sarcolemma on the myofiber, so it's very specific to the muscle cell. That region right there is called the end plate. Why? Because it's the region very rich in acetylcholine receptors, which are also ligand-gated channels for sodium ions. So it's a very rich region. It's actually not straight out, it goes in and out, basically to increase surface area for even more receptors being available. And it's called the end plate. So when there is depolarization at the end plate, it's called the end plate potential. It's a local depolarization. It's only locally right there where the axonal end is facing the sarcolemma right there. So basically in that region that I drew. So once this local depolarization has been produced and it reaches the threshold, remember we have to reach a threshold to stimulate the downstream voltage-gated channels that will open and allow more sodium ions to flood inside the myofiber and increase even more this depolarization. So now we go from negative 55 to we passed the 0 and we're into the plus 35 millivolts. So I'm going to write here in red plus 35 millivolts. So this is where we have gone across. We've completely depolarized the membrane and the charges are completely reversed, and that's why there's a plus sign here. All right. So there's the action potential. It got generated, and now we excited the cell. So we move on to the next phase, which is the action potential traveling down the sarcolemma. So we're still using red here, and a plasma membrane of the myofiber was already in red in the previous diagram, and we have an action potential that is traveling down this membrane. What that means is that there are more voltage-gated channels that open up like a domino effect, and we have these depolarization that are taking place as this is moving forward. So the depolarization with plus charges on the inside, negative charges on the outside, this is a depolarization. And it's moving along because more channels are opening, more sodium ions are coming in, and we are having depolarization that is propagated along the membrane. Now on the back side, we also have repolarization with potassium channels that are opening to allow for potassiums to flow back out and revert the polarity at the membrane to come back to resting membrane potential. But here we're talking about excitation contraction coupling. And what I want to mention now is the T-tubule. And I'm really exaggerating the T-tubule right here. This is a T-tubule. And this propagation is going to take place down the T-tubule. And I describe this as like a roller coaster ride. We have this depolarized membrane that is generated as this is propagated down the membrane here. So the action potential propagates down the membrane, and down here, if you remember just from learning the cell structures of a myofiber, inside we have the sarcolemma, which is the equivalent of a sarcoplasmic we have a sarcoplasmic reticulum, which is the equivalent of an endoplasmic reticulum right here. So this is a very elaborate structure of cisternae. So there's a lot of tubes that are interconnected and sacs. And so think of it as this big, this is all this is a closed compartment. So maybe I'll just close it here. And inside of it, right in here, we have lots of calcium ions that are basically stored in here. They're inside. This is a storage space for calcium ions. But we have voltage sensitive channels right here. And oh good, this just happens to be the same color I used in the previous slide for voltage gated channels. So these voltage gated channels are actually going to open up when they sense the arrival of the action potential of this depolarization at the membrane down here. So as this is coming down here, this is the depolarization. So we have a depolarized membrane over here. And this is going to trigger the opening of these channels. And when these channels open up, we have the exit of calcium ions. Calcium ions that are normally really highly concentrated inside of the sarcoplasmic reticulum, now is going to exit the sarcoplasmic reticulum and is going to end up outside of the sarcoplasmic reticulum. But we're still inside the cell, of course. This is all inside the cell. And what's going to happen is that we have our thick and thin fibers that are down here because we're inside the cell, and we have these thick and thin fibers of actin and myosin. So the thin fibers are actin and the thick fibers are myosin. And this is just a I'm just illustrating both fibers. I'm not drawing a specific sarcomere here, but to show you the fibers are right here. And as you know, we also have regulatory proteins of troponin and tropomyosin that are here. And troponin is the one that's going to bind to the calcium available now in the sarcoplasm. So I had described how troponin, upon the binding of calcium, this complex troponin-tropomyosin shifts. So there's a conformational change. We talk about a conformational change where the whole structure, the whole shape of this complex changes and moves out of the way to reveal binding sites on the actin filaments, and these binding sites are for the heads of the myosins. So this is basically the stage here is this particular part of the story is how we have excitation, contraction, coupling. So how do we have an excitation on the surface of the membrane? How do we let the contractile fibers inside the cell know that there's an excitation that arrived from a neuron, right? The neuron's coming to tell the muscle cell it's time to contract. How do we let the fibers inside know that it's time to contract? It's via this roller coaster ride down the T-tubule. So this is going to be called excitation, contraction, coupling. And in the next video, I'm going to explain how we have the actual contraction that's going to now take place. Now that we activated the contractile proteins, because the regulatory proteins, troponin and tropomyosin, are now out of the way because of the presence of calcium, now we can finally do a contraction. And there's going to be that whole sliding filament theory that's going to take place next. So that will be the contraction phase in the next video.